Dr. Amit Awasthi is Associate Professor at Translational Health Science and Technology Institute, Faridabad. He received PhD from NCCS Pune and has number of years postdoctoral experience. Dr. Amit is a renowned scientist and with very excellent publication, and he received number of awards, namely GP Talwar Young Scientist Award, Travel Award, Ramlinga Swami Fellowship from DBT, Innovative Young Biotechnologist Award from Department of Biotechnology, Young Scientist Platinum Jubilee Award in Biological Science, AAI Early Career Faculty Travel Grant Award, GP Talwar Mid Career Scientist Award, and Early Career Award from Dutch Society of Hymnology. And recently, he has been elected as Yes Ramchandran National Bioscience Award for Career Development 2020 and 21. So, Dr. Awasthi will deliver the talk today on secondary signal pathway in immune system and its function. So, I invite you, Dr. Awasthi. Please, over to Dr. Awasthi now.
Thank you very much for the introduction, and just let me share. So you can see my slides.
Yes, slides are visible.
Okay.
Move ahead.
Where is? Okay. So and I really thanks for the society for inviting me for the lectures. And I'm going to briefly discuss about secondary signal pathways in the immune system and function, and it's going to be more fundamental and basic kind of talk. And then at the end, I will try to talk about some of the work which my lab is actually being doing. And so basically, and we have got the introduction about the kind of cytokines, the T cells, and then Girdhari has talked about chemokine receptor signaling, and in context of various disease. So we have some idea about immune system. So basically, the first question what we should ask, and the student particularly should ask, why do we need T cells? Because I think majority of the work can be done by the B cells, and the B cells produce a lot of neutralizing antibodies, and people have now seen the importance of these neutralizing antibodies in various diseases, particularly respiratory diseases like influenza, and very recently in the COVID pandemic. So basically, if you have an infection and you see a lot of soluble proteins, which you can see, and these soluble proteins can be bind to the antibodies, which is also soluble, and they are produced by the B cells or the plasma cells, and these antibodies goes into the blood and the serum, and therefore they actually neutralizes the virus by binding to their proteins, and that's how either clearing the stopping the viral entry into the cells, or it is actually also fixing the virus through the activation of complement pathway. So basically, B cell has a big role, and I think majority of the things can be done. Then why do we need T cells? So one of the things which B cell cannot really do is actually look inside the cells, because some of these intercellular pathogens, virus as well as bacteria, they goes inside the cells and they live within the cells and replicate themselves. So the B cells can only see them outside when they're outside of the cells, but when they're inside hiding themselves, the B cell cannot identify them. So therefore, the T cell has the capability to look inside the cells through their T cell receptor. And one of the difference between the B cell and the T cells, the T cell actually identify these proteins or antigens, rather I should say antigen, in a kind of bound forms. So basically, if a and bound form as well as the linear form. So B cell can identify a protein in a kind of in 3D form, but you know that the T cell receptor can identify the antigen from these infected cells in a linear form, in a peptide form, and that too in a bound form through the MSC class 2, as you can see on this infected cells. So therefore, in order to find these hidden pathogens, definitely we need T cells. The second thing what T cell does is actually help B cells to do class switching, affinity maturation. So if you don't have T cell signal to the B cells, the B cell primarily keep producing IgM, and you know IgM is not really the antibody which we are looking for in the longer term immune response. So therefore, this IgM to IgG switching or IgM to IgE switching, right? You need a T cell signal to the B cells. So therefore, T cells comes in the central, and as a result, it not only identify these the cells which contains the virus and kill them. At the same time, it also actually help evaluation or evolving the B cells to produce more high affinity antibodies. So basically, this is the rules of adaptive immune system from the T cell point of view. Developing T cells mature and are tolerized in the thymus, in the central lymphoid organ. As we know that the T cells mature in the thymus, and the T cell which are actually reacting to the self antigen are being deleted in the thymus. As a result, it's called central tolerance. And the T cells which actually comes out from the thymus are actually normally, in normal case scenario, recognize only the foreign antigen. There is a certain percentage of T cells which actually comes out of the thymus are reactive to the self antigen, but those are very, very small amount, right? And when this small T cells frequency, which is a self-reactive to self antigen, when they comes to a situation where you have the environmental cues as well as you have kind of more recurrent inflammation, so your self-reactive T cells can amplify, and as a result you actually end up having autoimmune disease. But the normal scenario, the T cells are there not to cause any autoimmune disease, is just to protect us from the pathogens. So naive T cells, when they come out from the thymus, they goes into the secondary lymphoid organs, and Girdhari has very nicely mentioned about the role of chemokine and chemokine receptor, while the T cells actually migrating or traveling from one place to other place. And in the secondary lymphoid organ, primarily the lymph node, uh, in search of antigen. So when the T cells are actually coming, which is a virgin T cells or the naive T cells come out from the thymus, they go to the secondary lymphoid organs like lymph node and the spleen, and they try to find the antigen in order to, without the antigen, they cannot really survive longer. So therefore, the turnover of T cells is actually very high. So if they don't find the antigen, they eventually die of apoptosis, and then the new lot of T cells actually come from the thymus and again repopulate the secondary lymphoid organ. And this is the cycle actually goes on. At the time they find the antigen in the secondary lymphoid organ, they clonally expand them, because the frequency of each of these T cell receptor specific T cells for various antigens are very small, very minimal. So therefore, in order to amplify, they need a signal, and they amplify, clonally expand, and then they migrate into places where they have to do their job. So T cell require, basically, in order to do this activation or proliferation or clonal extension, T cell requires two signals for activation in the secondary lymphoid organs. And the moment they get these two signals, the primary signal is coming from the T cell receptor bound from the MHC class 2 peptide. And the second signal is costimulatory signal, which I will go into more details in the next slides. And then these T cells get activated and they become effector cells. And these effector cells migrate out of the secondary lymphoid organs. And this migration again require a lot of chemokine gradients. So one of the chemokine receptor for this important job is CCR7. So depending on the CCR5 and 7 combination, these T cell actually goes out of the lymph node, and then they go to the target organ where they find the infection, right? And that's how they start killing them. After, when they amplify the eradication of infection, then there's a phase of contraction, and you can see that in this slide very nicely. So you have infection, early infection. Within 0 to 7 days, you can see the infection is there, and during this time you have the T cell priming in the secondary lymphoid organs, and during this priming the T cell receive both primary and the secondary signal, and the T cells, both CD8 and CD4, they expand clonally, and then you can see the number from like 100 cells, it goes really very high, and it can reach to like million. And these are clonally the same cells, so they express all same T cell receptor, and now they are kind of in a form of army, and they actually ready to bang on those infected cells. And once they actually do finish their job, they cannot really survive longer, because then it can cause more collateral damages, which we really want to avoid. So therefore the T cells is having the intrinsic mechanism to, after they induce their effector function, after they finish or complete their effector function, they have to contract, and during this contraction most of these effector cells actually die in a process called activation induced cell death, and some of the cells actually remain in the body, and they remain in the form of memory T cells, and these memory T cells actually survive and actually migrate further into different places, particularly in the bone marrow. And when they see the next time the same antigen or same infection, these memory T cells can very quickly divide and clonally expand, and again come back to the target tissue and finish the job, contract again, go back. So this is kind of a back and forth cycles actually happens in the T cells, clonal expansion, effector, as well as contraction. So this can be, you can see, visualize this here. This has taken from the Dr. Shiv Pillai and Abul Abbas book, and you can see when the T cell required to get activated, or T cell need an activation, it has to be activated through the antigen presenting cells. And when we talk about the antigen presenting cells, and I'm referring this to here as a professional antigen presenting cells. So these dendritic cells are the cells which is actually called professional antigen presenting cells, because they express very high levels of MSC class 2 molecule, and they also express some degree of costimulatory molecule. So basically these dendritic cells lives in the under the skin, they called Langerhans cells, or they stay in the dermis, and they call dermal dendritic cells. So once they actually find an infection, or pathogen, rather should I say pathogen, these pathogen actually goes and interact with the dendritic cells, and dendritic cells is also having a kind of a phagocytic function. It is not the professional phagocytic cells like macrophage, but dendritic cells is having a phagocytic function to some degree, and as a result the pathogen can be engulfed by the dendritic cells, and the moment dendritic cells engulf these pathogens, there's a signal for dendritic cell to migrate. So then the dendritic cells start migrating, and during this process of recognizing the pathogen, it is all the pathogen also gives them the danger signals. So there is a danger signal in the pathogen through in terms of what you call LPS and other TLR activating molecules and the pathogens. And these danger signals ask the dendritic cells to express another receptor, which is called costimulatory molecule. And once the dendritic cells has the two of them, one is the MSC class 2 together expressing the peptide of that pathogen, and then during this danger signal, the costimulatory molecule is actually being expressed. And now this dendritic cell is now potent antigen presenting cells. It is having two signals, and then it migrates to the lymph node. And in the lymph node, it actually find the right T cells which recognize this peptide. So this is very important. And once they find the right T cells, the T cells start getting both the signals, and then T cells start clonally expand. And after this, the T cells actually move out. So you can see further here, the dendritic cells actually having the signal, both class 2 as well as the costimulatory signal. it actually activates the T cells. T cells get proliferated and we call them effector T cells. And this effector T cells actually goes out to the site of infection, and at this place the actually activation of effector T cells, at the site of infection eradication of the microbe. So this is how the dendritic cells and antigen find specific naive cells in the secondary lymphoid organ, and in the T cell zone activated effector T cells return to the site of the infection. So this is how the primary uh, function of dendritic cells in the T cells in context of co-stimulatory molecules. So basically what we really refer to the co-stimulatory molecule. So we can say co-stimulatory molecule, or we can say secondary signal, this is kind of synonyms, and which actually activate the immune cells. And this activation is done through the antigen presenting cells. And T cells require two signals to become fully activated. In case of first signal comes from the antigen through the T cell receptor, and the second signal actually is the co-stimulatory signal, or we can say secondary signal, is an antigen non-specific. So the secondary signal is always non-specific, and it can through the TLR pathway or NLR pathway, the secondary signal can be activated, and it comes on the expression of the secondary signal on the cell surface comes, and therefore it provided the interaction between co-stimulatory molecule expressed on the membrane of APC and the T cells. So one of the best characterized co-stimulatory signal molecules expressed on the T cells is CD28, and the CD28 which expressed on the T cells is interact with the CD80 or CD86. We also call them B7 family molecules, and we call them B7.1 for CD80 and B7.2 for CD86. The CD28 in general and normally on the T cells are expressed constitutively. So the expression of CD28 is more or less stable. It is not inducible. However, the CD80 and 86 on the antigen presenting cells are inducible, and these CD80 and 86 expression or B7 molecule expression on the APCs as actually induced by the danger signals, right? Other co-stimulatory molecule which is also kind of well characterized in case of T cells, which is a CD40 ligand and ICOS, which is expressed on the T cells, and they interact on the antigen presenting cells with CD40. So CD40 ligand actually interact with the CD40, and ICOS is actually interact with ICOS ligand on the APC. So basically, once this signal, so first and second signal, which is a antigen presenting, MSC TCR signal, and second one is a CD28 signal. The moment this two signal actually goes into the T cells, the T cells start expressing in a sequential manner the CD40 ligand. So CD40 ligand on T cell is an inducible ligand. It is not constitutively expressed like CD28. So CD28 signal is important for T cells to express CD40 ligand. The moment CD40 ligand expressed, it interacts with the CD40 on the antigen presenting cells, and CD40 ligand and CD40 interaction tell the antigen presenting cells to produce some of the cytokines like IL-12, right? And this IL-12 actually gives signal for the T cells to become Th1. It also gives the signal to the T cells to proliferate. The second important thing happens during this interaction, and these interactions are actually, you cannot really say which one. This is like happened very quickly, and therefore it's very difficult to segregate them. So during this co-stimulation, the other signal is actually given to the T cell is a survivor signal, and the T cell start producing interleukin 2, and this interleukin 2 actually is actually the growth factor for the T cells, and then T cells become kind of proliferate because of IL-2. So T cell co-stimulation is necessary for T cell proliferation, differentiation, and survival. So if you don't have secondary signal or co-stimulation, the T cell may not be activated, and they will remain in a state called energy, T cell energy. And if you have T cell energy, this T cells can be also deleted, and in a longer run, it's called immune tolerance. So basically, even you have an antigen, and if you don't have the secondary signal, the T cell will be non-responsive, and that's what happens in case of autoimmune diseases. Why? Because every one of us is having some frequency of self-reactive T cells, which is coming out of the thymus, because not every machine is not really perfect, so nothing is ideal. So basically, some of these self-reactive T cells come out of the thymus in the periphery, but we don't get autoimmune disease as often, right? Because the secondary signal is not given, right? And the secondary signal is not given, that the MSC, self MSC containing peptide is interacting with a T cell, but T cell is not going to proliferate, and as a result, they will remain unresponsive and maybe deleted. So this basically, so the whole thing you can understand by this picture, that foreign antigen without danger signal. So you have a foreign antigen, which is actually being processed by the dendritic cells, and it is being expressed by the MSC class 2, and this MSC class 2 presenting this peptide to the T cell receptor, and this is a signal 1. But in this case, nothing going to happen. The T cell will not proliferate. T cell tolerance will happen, and T cell energy, T cell deletion, you can say. And as a result, you are not having any T cell response. But the problem, what happens really in case of foreign antigen, when the foreign antigen also contains the danger signal, or you call it microbial PAMs, right? So this microbial PAMs actually recognized by the toll-like receptors, or NLR, as a danger signal. And during this process, the foreign antigen will present through the MSC and the T cell receptor, that is signal 1. And then this co-stimulatory molecule are being expressed because it's induced by the toll-like receptor or a microbial PAM, and they interact with the co-stimulatory molecule C28. And now the T cell got 2 signal, and as a result, T cell will proliferate, produce cytokines, and that's a progressive T cell response. So you can also see that this very similar thing, what I have mentioned in the previous slide, the role of co-stimulation in the T cell activation. So first stage is antigen recognition. So MSC bound peptide is being recognized. The T cells, as I told you, constitutively express C28, right? But there's no response. And then you have the second situation in this activation of antigen presenting cells by microbes, innate immune response, right? And you can see now the activation of this APCs, now they have the second signal coming out, which is a B7, and they also produce interleukin 12. And as a result, you have the T cell which proliferate, and the proliferating T cells produce tons of IL-2. And because of IL-2 is a proliferative cytokines and T cell growth factor, you have effector T cells proliferation and differentiation. So as you can see from the similar thing from this, it's very nice 2 signals theory and cytokine production. And then you have a second thing, which is a T cell tolerance, because the second signal is not there. So what happens in some of these pathogens like HIV or intracellular pathogen, what they do, they try to actually hijack the second signal. So what happens when the microbes actually infect the macrophage or antigen presenting cells? What they do, they downregulate the B7 signal, right? So they try to produce a cytokines like perhaps IL-10 or other factors by which the B7 molecules get actually downregulated. The moment B7 molecule get downregulated, the secondary signal is impaired, and as a result, you have no cytokine production, even though in the infection you want to have this cytokine production in order to kill the pathogen, but the pathogen is trying to outsmart the T cells, and they try to impair the second signal, and as a result, they can survive very nicely within the cells. So the second thing is in the immune system is also, immune system is having an inbuilt capacity to self-regulate, because if you don't have the capacity to self-regulate, the immune system will go haywire, and as a result, rather than killing the foreign pathogens, immune cell will do more collateral damage, which is going to be dangerous for the individual, and as a result, individual may come to death, right? So therefore, immune system has this inbuilt regulatory mechanism, and one of the mechanism is the CTLA4. So basically what happens, the CTLA4 normally within the T cells And when the T cells get activated through the primary signal, through the T cell receptor, and the secondary signal through the CD28, now immune cells are getting activated, but at the same time, immune cells or the T cells are actually telling to the T cells, now the T cell need to stop their proliferation. T cell need to stop because too much proliferation will be detrimental for the host. So what happens? The CTLA4 comes on the surface, and the CTLA4 and CD28, they have the both having the affinity to B7 molecules. In fact, the CTLA4 is having more affinity to the B7 molecules. So as a result, the CTLA4 bind to B7, and then the CD28 signal get interrupted. And again, the T cell which were proliferating very fast say now stop. So this is the kind of inbuilt self-regulatory mechanism. So when this secondary signal were being identified, at the same time there are other secondary molecules or secondary signal which is a more into co-inhibitory receptor also being identified is PD1 and PDL1. So PD1 is expressed on the T cells, and actually the experiment has been done by the Rafi Ahmed group. So basically Rafi Ahmed group was doing, they were putting like a chronic viral infection to the mice, and when you put the chronic viral infection and when the chronicity increases, the T cells actually start expressing this inhibitory molecule called PD1, and then once the T cells start expressing a PD1, the T cell will stop proliferating, and at that time Dr. Rafi Ahmed's group actually called this process or phenomena T cell exhaustion. So T cell are exhausted if they're expressing PD1, and if you somehow break this exhaustion by actually blocking PD1 or PDL1 by anti-PD1 antibodies, the T cell again will start proliferating and can clear the virus. So this is how initial has been identified, and later on the similar thing has been shown in case of tumor immunology. And based on this, the CTLA4 and PD1 therapy is actually now working in the tumor setting, because when the T cells actually are there and go to the tumor setting, rather than they start proliferating, the tumor cells is giving a negative signal to the T cells, and these T cells start expressing a lot of PDL1 and the CTLA4, and as a result, the T cells which is supposed to kill the tumor, they are now become exhausted and sitting in the tumor and not doing anything. And therefore, if you block CTLA4 and PD1 in the tumor environment, therefore the T cell become again activated and start killing the tumor. So as Girdhari very nicely talked about some of these molecules, and this is, I'm giving you a kind of nutshell that other than the MSC and T cell receptor signal, what I also talked about B7 and CD28 signal, you can see here, and the same thing, B7 can also bind to CTLA4 because it is having more affinity. The other signals, which is I talked about briefly, CD40 on the antigen presenting cells and CD40 ligand on the T cells. The other signal is like LFA, which is very important because when the T cells and the antigen presenting cells are interacting, you need some sort of stability because this interaction is very dynamic. So in order to have this stable interaction, you have to have this adhesion molecule, what is called ICAM and the LFA. And this adhesion molecule gives the stability of the T cells and the APCs and to make a kind of stable synapse in order to activate the T cells. So one of the important things is also kind of essential by the secondary signal. You can have the two pathways of signaling. One pathway you can see on the right hand side is actually T cell receptor, and this is a proximal event of signaling what T cell receptor does. And then the other signaling actually coming from the CD28 through the V7, and the CD28 actually activate the PI3 kinase pathway, and PI3 kinase pathway is important, is called the survival signal, and this PI3 kinase again goes and activate the other signaling component and Web1, RAC, and so you have kind of multiple different kind of effector signaling molecule. One is the PKB-AKT pathway, which gives the survival signal to the T cells. The other one is the MAP kinase or SAP kinase or GNK pathway, which is important for chemokine and other cytokine production. And the third pathway, which is a PKC pathway, merged into the NF-kappa B pathway. And this NF-kappa B again produces different cytokines of Th1 and Th2. And now the role of NF-kappa B also extended in terms of a newly discovered subset of T cell of Th17 and Th9 and other subsets, so on. So this is how the secondary signal is so crucial, not only for the survival, but also for the chemokine, cytokine production, as well as other co-stimulatory molecule expression. So ever since the discovery of these co-stimulatory molecules, now the co-stimulatory molecules can be identified or actually divided into two categories. And this discovery after the tumor immunology field actually evolved about the co-stimulatory and co-inhibitory molecules, and the advancement in case of sequencing and other like CyTOF and various techniques. So by which now we have many co-stimulatory molecules. You can see some of them in the green, they are the activators, and you can see some of them are in the red, they are the co-inhibitory molecules. And why we call them co-inhibitory or co-stimulatory? Because particularly co-inhibitory, because they are actually expressed in a cohesive fashion. So it is something that when the CTLA4 will express, the same cell will express PD1, and sequentially they will express TIM3. So it is not like one co-inhibitory molecule will be expressed on the one T cell. No, they come in a kind of a cohesive manner together, and that's how they actually able to have profound inhibition of T cell proliferation or T cell function. So therefore, if we know some of these molecules in a more details and their ligands, so therefore now people are talking about not single therapy. They're talking about we can put like PD1 CTLA4 together to have better T cell response, or we can put PD1 CTLA4 TIM3 together to have even more better response. So once we identify some of these dominant co-inhibitory molecules, and so we can really neutralize them together in order to have better T cell response in case of tumor immune response, rather than neutralizing only one and one. And example has been shown in the clinical trials, like PD1 and CTLA4, when you do combine, they actually do better anti-tumor immune response rather than alone in tumor immunity. So again, I just put again the same thing, how the inhibitory molecules actually have interplay. So basically you have antigen presenting cells. They are actually doing antigen presentation to the T cell receptor, and obviously then the secondary signal through C28, you have PD1 as well as CTLA4. So this side is actually more into the antigen presenting cells. But when the T cells actually interacting with other cancer cells. So cancer cells express definitely MSC class 1 having cancer antigen, and cancer cells express some of the other inhibitory as well as co-stimulatory molecule, which is not being expressed by the antigen presenting cells APCs. So that's how kind of there is an interplay between the T cells in the lymphoid organs, as well as when they are out of the lymphoid organs, they go to the cancer site or the target tissue, they have to interact with different kind of co-stimulatory and co-inhibitory molecules in order to achieve the optimal function. So I think one of the important thing, how this co-stimulation and co-inhibitory molecules actually tells the cells that you have to either proliferate or stop proliferation. So basically there are two different kind of signaling moieties has been described and identified. One is called ITAM, and this is the activation motif. This is a tyrosine based activation motif, and they actually give the progressive signal to the T cells to induce activation of the immune response. But when we talk about this inhibitory receptor, they activate the ITIM motif, which is a tyrosine based inhibitory motif, and this inhibitory motif again gets phosphorylated and stops signaling and tell the T cells to stop proliferation and activation. So this the interaction of ITAM and ITIM is also very crucial in order to fine-tune the T cell response in various settings. So basically, now I quickly go on to some of the work which we were doing in the lab, and some of the work which we have published, and some of the work which in press, and I'll just briefly talk about. We know Th9 cells has recently, I mean not recently, almost 10 years ago, we have identified a new subset of T cells which is called Th9 cells, and these T cells produce interleukin 9, and the role of interleukin 9 and Th9 cells has been now described clearly by various investigators after we identified these cells. So they are very important potent cells in tumor immunity. They are also important for clearing extracellular pathogens like Helminth infection. And the role of Th9 cells has been also shown into the ulcerative colitis. So they are primarily effector cells in the ulcerative colitis. So somebody is having more ulcerative colitis, or it has been shown in the UC patient as well as in the mouse setting, mouse oxazolone-induced colitis, that in the colon, the lot of accumulation of Th9 cells or IL-9 is there that causes the inflammation. The role of IL-9 has been shown in the allergic inflammation because IL-9 has been shown to be associated with asthma, because if you people have shown that the IL-9 in the lung, or they have created a mouse in which the IL-9 is produced transgenically in the lung epithelial cells, and these mice actually have a lot of mucus production and they have asthmatic or breathing issue. So IL-9 has been shown there's a very crucial role in the asthma. So basically, having such importance of IL-9 and Th9 cells, we wanted to identify what are the transcriptional factors or transcription regulation of Th9 cells, and we did the RNA-seq analysis, and I'm not going to go too much detail into it, and we identified one of the factor called FOXO1, and this FOXO1 is important for IL-9. It actually bind to the IL-9 promoter and activate IL-9, transactivate IL-9 and produce more IL-9. And you can see what IL-9 is also produced by the other cells than the Th9 cells. And we found the FOXO1 is definitely important for IL-9 production in Th9 cells. FOXO1 also important for IL-9 production in Th17 cells. But at the same time, FOXO1 suppresses IL-17. So it is actually a kind of switch between the IL-9 and IL-17 Th17 cells. And very similarly, we have shown that the Th2 cells produce IL-9. But the IL-9 production between the Th9 cells, Th17 cells, Th2 cells, and iTreg cells are variable amounts. So highest amount of IL-9 is being produced by the Th9 cells, then the Th17 cells, then the Th2 cells, and then the Treg cells. So my lab wanted to understand whether FOXO1 is actually regulating IL-9 production across the T cells. So that's what we have also found, that when you have more FOXO1 in Th2 cells, it produces IL-9, and it really does not do too much to IL-4, 5, and IL-13. And in case of FOXP3 cells also produce IL-9, and FOXO1 is important for that production. And FOXO1 is also important for inducing suppressive function of FOXP3 positive cells. So we have published these results. And during this analysis, we were also identified some of the pathway which is important, and we found one of the pathway called EGFR receptor expressed on the Th9 cells in our RNA-seq analysis. And we found that by the real-time PCR, we confirmed the result that Th9 cells produce a lot of EGFR. And using the EGFR conditional knockout, where we have deleted the EGFR in the CD4 pre cells, and then we try to differentiate the Th9 cells. And when you don't have EGFR, the Th9 cells are not being differentiated as compared to wild type. So we are saying that Th9 cell EGFR is important for Th9 cells. So basically, I apologize. I think there is a kind of this black circle. It was not supposed to be black. It is it's called amphiregulin. So EGFR is having 6 different ligands. You can see 6. I mean, there are 7 different ligands, including the amphiregulin, which is now a black circle. So these different ligands of EGFR are expressed by different tissues, and amphiregulin, which I have made the red circle, is expressed by the T cells. And therefore we focused on the amphiregulin, but just to find whether amphiregulin interaction with the EGFR is important for Th9 function and differentiation. So what we have used amphiregulin knockout T cells. So we have taken the wild type Th9 cells and we have taken the amphiregulin knockout Th9 cells, we differentiated them. And then we did the RNA-seq analysis, and we found that the IL-9 is actually being suppressed or inhibited when you don't have amphiregulin, and as compared to the wild type, which is a red color. So this clearly says that EGFR pathway through the amphiregulin is actually important for IL-9 production. We further went into the detailed RNA-seq analysis, and we found various, and we focused on various transcription factor, as I have shown in case of FOXO1, but we have found additional transcription factor which are also important for Th9 differentiation. And as you can see in this slide, the transcription factor which I call upregulated in Th9 cells, and the transcription factors are downregulated in Th9 cells. So we focused on the positive regulator in case of transcription factor in Th9 cells, and we found HIF1 alpha is upregulated in Th9 cells. And our analysis in the IL-9 promoter, we found HIF1 alpha sequence is there in the IL-9 promoter, and we did the luciferase assay, and we found when you actually put the HIF1 alpha together with the IL-9 promoter in the luciferase assays, you can find that HIF1 alpha actually transactivate IL-9 promoter. So this paper is just got, in principle, got approved, and it's going to be out soon. It's going to be out soon, very quickly. So basically, we have done a lot of experiment in that paper, and I'm just trying to summarize that in a kind of a form that everybody can understand. That what initially we had identified in 2008, that you need a 2 cytokine signal, TGF beta and IL-4, which bind to TGF beta receptor and IL-4 receptor. And this signal actually, now we connecting more dots, we identified that this signal actually activate the EGFR, and this EGFR comes on the surface of the Th9 cells, and this EGFR actually bind to amphiregulin, and this amphiregulin EGFR axis actually activate HIF1 alpha. And this HIF1 alpha, what it does, and we have done detailed metabolomics profiling, which is part of the this paper, and we found that succinate is actually one of the important metabolites for Th9 differentiation, is actually amplify or activate HIF1 alpha, and this HIF1 alpha bind to IL-9 promoter and activate IL-9, and these Th9 cells produce tons of IL-9, and that's how they do the tumor killing. So that's what we have shown in this paper. And this amphiregulin further amplify and activate this whole pathway. So I just quickly take a couple of more slides just to quickly show one of the paper which we have published last year. So my lab was focusing on a lot of gene expression analysis through the RNA-seq to identify various transcription factors. But there is a limitation of RNA sequencing, that you can actually only identify the genes which are inducible. You cannot really identify the proteins which are always there in the cells, or the proteins which are post-transcriptional or post-translational modification. So therefore we implied the proteomics analysis, and we did the TS0 and Th9 proteome analysis, and we did the analysis and found the proteins which are differentially expressed in the Th9 cells and Th0 cells, and some of the proteins which are common in these settings. And when we analyze this data, I'm sorry that the slide is too crowded, and you can see the red circle. So in the red circle, we identify, after the proteome analysis, the PP2A. There's a PP2A, one of the signaling molecules, which is a phosphatase. So we identify this PP2A as one of the important protein, which is important for Th9 differentiation in the proteome analysis. And then we try to understand how the PP2A is actually interacting with already known transcription factors of Th9 cells. So for example, we have identified FOXO1, and we have also identified HIF1 alpha, but other groups has identified IRF4, GATA3, SPI1, BATF, and IRF1. So these are the transcription factor has been shown having a role in Th9 cells. So we try to understand how this PP2A is interacting with these different transcription factor. And during this analysis, we found there's a there's a kind of clear-cut interaction of PP2A. So then we wanted to understand whether if we block PP2A, can we also block these transcription factors in gene expression? So then what we have done, we use LV100, which is a PP2A blocker, and we block the PP2A. You can see all these different transcription factor. What you see in the interactome is also inhibited, like SP1, IRF4, HIF1 alpha, BETF. So all these transcriptions in FOXO1 all are actually suppressed. So it looks like the PP2A is actually controlling one of the major signaling node, and this signaling node is important for all the transcription factor, which is important for Th9 cell generation, are essential, and therefore this is an important pathway we have identified. And then we finally tested the role of PP2A in the tumor environment and tumor setting. So what we have done, we have actually taken the B16 melanoma model, and then these B16 melanoma cells are expressing OVA as an antigen. So these cells can recognize, they express OVA, and the T cell can recognize this OVA. So we have used Th9 cells, which is the OVA specific Th9 cells. So then when we injected Th9 cells, which is OVA specific, and the tumor which is expressing OVA, so you can see red line actually suppressed. The tumor burden is suppressed to green line when the Th9 cells, OVA specific Th9 cells, actually killing the tumor. And when we block the PP2A, the effect of anti-tumor effect of Th9 cells actually impaired or abrogated, as you can see by the tumor growth and the tumor weight. So I think with this, I really would like to thank everyone, and I will be happy to answer any question related to the work what I'm doing in the lab, or any related to the first part of the talk where I talked about secondary signal and co-stimulatory molecule. Thank you very, very much.
Please unshare the slide, Dr. Amit. Yeah.
Hello.
So now session is open for the questions. So I don't have any questions for the panelists.
I can stop sharing the slide. Yeah.
One of the issue is actually, is there any way we can try to balance out between the positive signal, like for the antigen stimulation, versus the negative signal for the antigen recognition? Because if there is a way to balance out, we would be able to manipulate either the autoimmune disease where we want no response, and in the tumor immunology where we want higher response. Because what we face in tumor, when we use anti-PD-1, we start getting autoimmune disease. And in the same time, when we try to immunosuppress with the inhibitors, we are moving towards the development of few increased incidence of malignancy. Is there any way to understand this balancing process?
So I think one of the feature of the immune cells, or it's a double-edged sword. So you can use for tumor, and if you try to suppress tumor, or you try to kill tumor, it will have more autoimmune disease. Like CTLA4 is the best example. I think in order to do this, maybe we need to focus on the co-inhibitory signals, which is not as robust. Like CTLA4 is kind of very robust signal, right? So for example, the toxicity when you see in the tumor patient, when you do anti-CTLA4, in terms of IBD, you get a lot of inflammatory bowel disease in those patients. But when you neutralize PD-1, the toxicity actually goes dramatically down as compared to the T cells. So therefore, maybe I think that each tumor, we need to screen or sequence or whatever the way by new techniques to identify what are those specific inhibitory molecules are being expressed. And then we should focus on the molecules which are not that robust. However, they are selectively more expressed in the tumor setting, right? So I think this may be the way to say that in this tumor, in this patient, and this will again become like more personal medicine, that in this patient, the PD-1 is more expressed, or in other patient, the ICOS is more expressed. So let's focus on ICOS in this patient versus PD-1 or the other. So I think this personalized way to do will be much better, rather than tweaking the way you were saying. Is maybe at this point of time bit challenging and difficult. Maybe the new techniques will evolve in the future, which will give us an idea how to do this kind of thing.
So thank you, Dr. Amit. Very excellent.
Can I ask one question?
Yeah, sure.
Yeah, Amit, it's very good talk, very nice data. I just wanted to check, have you checked the Th9 in asthmatic or other allergic patients?
We haven't done in the patient, but other groups has been shown about their accumulation in the asthmatic patient. And there was a company called MedImmune in the Washington DC. They were having the anti-IL-9 in the clinical trial for asthmatics. However, in our first paper, we have done the asthma as a model, and we have shown that when you block FOXO1, you block IL-9, you have better response. There is a less asthma in the mice. Yeah.
So is there a balance in Th9 and Th17 in those patients?
So I think this is kind of very interesting question, and I think there's a lot of sophisticated tools are required to address this question, because when we talk about pathogenic and non-pathogenic Th17 cells, I think this non-pathogenic Th17 cells does express IL-9, and so basically we need to have some way to identify whether the cells which goes, let's say that the role of Th17 cells were also being implicated in the asthma. But later on, there is a kind of confounding reports actually keep coming, whether to neutralize IL-17. There was kind of very contradictory results. But I think maybe some of the Th17 cells, the moment they move to the lung, they maybe start expressing the IL-9 together with them. So rather than neutralizing IL-17, we should neutralize IL-9 from those Th17 cells. I think similar thing in the gut also. IL-9 is also important for gut permeability. So I think this is an outstanding question, is still there, but we need to have more tools to identify what is the role of IL-9 in the Th17 cells.